Hi, my name is Michael Karras, and my topic was to examine the respiratory exposure of dinitrobenzene and its effects on the testes. Giving a brief introduction to the toxin, dinitrobenzene exists in its crystal form as a yellow powder, shown here. Here is the structure of dinitrobenzene, which we'll examine in more detail in a second. Dinitrobenzene is not naturally occurring and is synthesized in a reaction with benzene, nitric acid, and sulfuric acid. It is commonly used as an intermediate in organic synthesis and is used to manufacture dyes, plastics, and explosives. It often exists as an impurity in the production of tinitrotoluene, TNT. Since dinitrobenzene is a product of organic synthesis and is highly regulated, it is unlikely organisms will come into contact with it. However, occupational exposure may occur through inhalation or dermal contact with this compound at workplaces where dinitrobenzene is used. Waste discharge from explosive manufacturing plants can also cause an increase in levels of dinitrobenzene in the air, land, and water in the surrounding area. Dinitrobenzene can be absorbed into the body by nearly every possible route, the most notable being dermal, respiratory, and oral exposure. By looking at the structure, protonatable groups, relative solubility, and some experiments in literature, the absorption of DNB will be analyzed. When examining the structure, the aromatic ring is extremely nonpolar and hydrophobic, whereas the nitro groups are extremely polar and hydrophilic. The nitro group even has ionic character, with a positive charge on the nitrogen and a negative charge spread between the two oxygen atoms. Even though the nitro groups are relatively polar, the hydrophobic properties of the aromatic ring have a much greater effect on the overall polarity of the molecule. The hydrophobic character facilitates its fusion across membranes. Since there is no protonatable groups, and therefore no pKa, it is highly probable that the molecule will be neutral as it reaches the alveoli. The log P of dinitrobenzene is 1.8, supporting the hydrophobic character. Dinitrobenzene is small and hydrophobic enough to make it past the nasopharyngeal and trachobronchial regions and enter the alveolar sacs. From the alveolar sacs, it must cross the alveolar epithelium, the fused basement membranes, and the capillary epithelium before entering systematic circulation. Other factors could also affect the bioavailability of DNB, such as steady-state kinetics, respiratory rate, blood volume, and blood clearance. When examining the distribution of dinitrobenzene, it is important to note that there is little experimental evidence outlining distribution following inhalation exposure in humans and animals. However, numerous indicators provide information about the distribution of DNB. It is well known that DNB interacts with the testes in mammals, so we know it gets to the testes somehow. Since DNB is hydrophobic and not very soluble in blood plasma, it is likely transported by plasma proteins which circulate the toxin throughout systemic circulation. Once absorbed in the lungs, dinitrobenzene will bind to plasma proteins and travel through the pulmonary vein to the left ventricle, where it will be distributed throughout the body. DNB will travel to the liver as well as the testes, where it is metabolized and exerts its toxic effects. It is unlikely that the toxin spends a significant time outside of distribution, since the side effects are limited to 48 hours after exposure. There is no appreciable concentration of toxin to remain in storage. Once absorbed, DNB must cross the blood-testes barrier between the lumen of the interstitial capillary and the lumen of the semi-infernus tubule. Plasma proteins can easily carry dinitrobenzene across the blood-testes barrier. Once dinitrobenzene enters systemic circulation, it will primarily be metabolized in the liver. However, many enzymes involved in phase 1 and 2 metabolism are also expressed in the lungs and Serodi cells in the male reproductive system. A few studies have shown that dinitrobenzene undergoes enzymatic and non-enzymatic oxidative and reductive biotransformations, followed by conjugation and excretion. A metabolic diagram from one of the studies shows that during phase 1 metabolism, dinitrobenzene undergoes a 2-electron reduction to form nitrosonitrobenzene. Nitrosonitrobenzene then undergoes another 2-electron reduction to form nitrophenylhydroxylamine. Nitrophenylhydroxylamine then undergoes a final 2-electron reduction to form nitroaniline. Now, one might wonder, why is metabolism making dinitrobenzene more hydrophobic? Shouldn't metabolism make toxins more hydrophilic to favor excretion? Well, the answer is that even though phase 1 metabolism is making DNB more hydrophobic, it's acting as a setup to precede phase 2 metabolism. Nitroaniline can undergo phase 2 metabolism and be conjugated to glucuronic acid. Nitroaniline can also be further oxidized or reduced to ultimately undergo sulfation or glucuronidation conjugation. Numerous enzymes have nitroreductase capabilities that are also able to reduce the nitro groups in DNB. These enzymes include xanthine oxidase, aldehyde dehydrogenase, and cytochrome P450. Since nitro groups can only be metabolized in anaerobic environments, we can gain insight as to where DNB will likely be metabolized. The gastrointestinal tract is a perfect anaerobic environment that has a high concentration of reductases, which favors nitro reduction, and the majority of metabolism will likely happen in the liver. Although nitroreductases are expressed in the lungs, the aerobic environment prevents metabolism. It is not likely that any metabolism will precede absorption in the lungs. Here is another schematic diagram showing the phase 1 metabolism of dinitrobenzene. It is important to note that a toxic metabolite is generated called nitroxyl anion radical. From the nitroxyl anion radical, more reactive and radical species can be generated, such as superoxide. Metabolism affects distribution in the sense that prior to phase 2 metabolism, the toxin has increased hydrophobicity, increasing its ability to cross membranes. However, the conjugated metabolite is very hydrophilic, making it difficult to cross membranes and be further distributed. Metabolism affects excretion in the sense that a nonpolar hydrophobic toxin was transformed into a hydrophilic polar molecule that favors excretion. Once metabolized, the final conjugated metabolite will be excreted in the urine. The hydrophobic properties favor aqueous solubility, and the conjugated toxins will be effluxed into the glomerular filtrate by various transporters. Glucuronidated conjugates enter the glomerular filtrate via organic anion transporters, OATs. However, the same transporters can also reabsorb the same toxic metabolites. If metabolized in the liver and excreted through the bile ducts, the metabolite can undergo enterohepatic circulation, delaying ultimate elimination. This is a metabolism-dependent excretion due to the fact that dinitrobenzene must first be made hydrophilic and water-soluble prior to excretion. Studies also confirm that conjugated metabolites of dinitrobenzene are present in rat urine. Since the parent toxin is different from the effector toxin, dinitrobenzene must first be metabolized before exerting its toxicity. The effector toxins are the nitroxyl anion radical, as well as various other types of reactive oxygen and nitrogen species. The target of the toxic metabolites and toxic byproducts of dinitrobenzene is the Sertoli cell of the male testes. Sertoli cells form the blood testes barrier, which creates the border between the semi-infirmous tubules and the interstitial blood space. Sertoli cells act as phagocytes, which consume residual cytoplasm during spermatogenesis. It essentially nourishes the sperm cells through spermatogenesis. Due to lack of research, the specific target of the metabolites of dinitrobenzene is still in question. However, the phospholipid bilayer of the membranes of Sertoli cells is likely affected. The mechanism of toxicity for the toxic metabolites of dinitrobenzene is not well understood. Numerous papers, some published as late as 2012, indicate a lack of knowledge of the toxic mechanisms. Most publications examine the toxic effects of dinitrobenzene rather than the underlying functional and regulatory mechanisms causing those effects. Examining the effects of toxicity can give insight into the mechanism. What is currently known is that a metabolite of dinitrobenzene causes apoptosis in the Sertoli cell. We should be looking for a mechanism that would cause target destruction or dysfunction that induces apoptosis. First, looking at the functional mechanisms, the formation of superoxide and nitroradicals will cause oxidative stress, leading to lipid peroxidation that causes cell death through necrosis. The superoxide radical will be further metabolized by superoxide dismutase and Vinken reactions to form hydroxide radicals. Hydroxide radicals pluck a hydrogen off an unsaturated lipid via hydrogen abstraction to form lipid radicals that propagate, leading to the destruction of the cell membrane and creation of mutagenic metabolites. Looking at the mechanism of toxicity from a regulatory perspective, numerous studies show that there is an increased expression of p53 in rats exposed to dinitrobenzene. p53 will translate proteins that cause a chain of activation leading to apoptosis. In addition to the initiation of apoptosis, there are other signaling pathways that are activated that lead to cell cycle arrest. Studies show that DNB leads to a downregulation of PLK, which has multiple effects on the cell cycle. First, it prevents activation of CDC25C, which in turn prevents activation of cyclin B, stopping mitotic entry. Secondly, downregulation of PLK prevents activation of APC, which is responsible for expressing CDC20, cyclin B, and PLK. APC will interact with CDC20, which ultimately causes the sister chromatid complexes to separate. Overall, a decreased expression of PLK will prevent sister chromatids from separating, halting cell cycle growth. p53 also halts cell cycle growth by activation of p21, which inhibits enzymes responsible for cell cycle growth. The regulatory mechanism of toxicity leads to apoptosis accompanied by G2 M-phase cell cycle arrest. The combination of regulatory and functional mechanisms of toxicity from dinitrobenzene lead to lipid peroxidation, apoptosis, and cell cycle arrest, ultimately leading to the destruction of Sertoli cells. Numerous studies confirm that the effects at the cellular level were regression of semi-inferior tubules that exhibited degenerated spermatocytes, formation of giant cells, and necrosis in Sertoli cells. In regards to effects at the organismal level, in rats, numerous studies show that dinitrobenzene will cause severe dysfunction and injury, harming the reproductive ability of the animal. In many cases, if given a high enough dose, the rats needed to be euthanized, implying severe bodily damage. A case study of 6 workers who inhaled dinitrobenzene reported cyanosis, palpitations, and an increased heart rate, but no testicular damage. The estimated LD in humans is between 5 and 50 milligrams per kilogram for a 70 kilogram adult. Dinitrobenzene's ability to induce apoptosis and minimize cell growth may be useful in developing chemotherapeutics.